It's my pleasure to introduce Professor Ruchi Anand, who obtained her MSc from IIT Kanpur in 1998 and completed her PhD from Cornell University, USA, and a couple of postdocs in USA and University of Pennsylvania and so on. Joined the Department of Chemistry in 2008, and currently she is a professor. And she has, of course, received several awards. And one of the main one is the Wellcome Trust DBT Alliance Senior Research Fellowship. And she is also elected member of National Academy of Sciences, Allahabad. And she is also editorial member of several journals that includes ACS Sensors, Asian Journal of Organic Chemistry. And she is also committee member of several funding agencies. She herself has a lot of fun. One can apply to her directly for funding. So with this short introduction, I just invite Professor Ruchi Anand to share her slides and present a talk.
First of all, thank you for the introduction, Professor Ravikant. And thank you to IIT Bombay for giving this award to me. I'm really grateful for IIT for acknowledging the contributions. And I really thank the jury and everyone for this award. So today I'm going to be talking about some of the research in our lab. In the interest of time, I will only be sharing a portion for which I had put forth this application for. But in general, our lab works on two basic areas. We work in development of biosensors, and here we look for develop biosensors which are protein and synthetic biology-based biosensors for detecting organic pollutants such as phenol, benzene, toluene in water. And we can go down to drinking level limits, and we have now been trying to shift into developing devices. We also do a lot of work in trying to understand the origins of antibiotic resistance. Basically, the question we want to answer is, why do this antibiotic resistance and drug resistance arises, and can we do something to combat it? What can we do to reverse resistance? Because we can't keep making new and new antibiotics on a daily basis, but what we can do is try to understand why bacteria who were okay with these antibiotics are becoming resistant. Can we stop this resistance from facilitating itself? What are the mechanisms which are governing it? So this is because drug resistance is a very, very silent epidemic, and it is thought that 300 million people may succumb to it by 2050. You may have so many deaths because drug resistance is becoming a huge issue. Simple infections, if you can't treat them in this world, then we will be in a very bad state as time progresses. So what happens is, most of the antibiotics that are produced, they are actually produced from soil bacteria, which are streptomyces. These bacteria have the resistance mechanisms in place. They produce the antibiotics. They know how to also prevent these antibiotics from killing them. So we also looked into streptomyces to understand the resistance mechanisms so that we can combat it. Here, in this regard, we had two major themes that were running in our lab. One in which we were looking at efflux pump regulators, and this work we did, a lot of this work we did, and I had mentioned this work, but in the interest of time, I will not be able to talk about this protein DNA complex. This particular complex helps in making doors. For example, when a bacteria is there and antibiotic pressure is there, it creates a lot of doors, which are efflux pumps, so that antibiotic is flushed out, so that the levels of the antibiotics go down. This particular protein that I'm talking about, which we worked on before, was involved as a controller of this door. It was like a key to the opening and closing the door. So we try to understand this, and if we can modulate this key, then we can basically develop inhibitors such that the key never opens. Then we can have high concentrations of antibiotics. Another mechanism by which drug resistance is propagated is via by modification of this machinery called the ribosome in our body. So this ribosome is the main machinery in our body. I'll have to change the laser pointer, sorry. So the ribosome is the main machinery in our body which synthesizes proteins. So what happens in the ribosome is that it synthesizes this protein via this machine. It reads RNA, and then from the side comes out the protein which is folded. Now, if you want to kill a bacteria, you would like to have antibiotics such that this protein production of the bacteria is stopped, not yours, only of the bacteria. And that's why most of the antibiotics, if you see, are antibiotics which stop this production of this bacteria. So this is the ribosome. And all these antibiotics, you must have taken streptomycin, erythromycin, tetracycline, and macrolides here I'm talking about, which is the main feature. They're common antibiotics which are used, where all of these go to the bacterial ribosome, stop protein synthesis. Now, protein synthesis is the main thing where all functions in our body in bacteria, they are the workhorses of the cell. If you stop that, then the bacteria will die. So you try to make selective antibiotics or drugs, as we call it, which will only stop the working of the bacterial ribosome. In this regard, there is this class of antibiotics called the macrolides, lincolnomides, and streptogramin. And the way they stop the protein synthesis is, you saw protein was coming out from the ribosome, and there was a tunnel by which the protein will come out. You put a plug in that tunnel. That is the antibiotic. So you do not allow the protein to come out. And if the protein cannot come out, it cannot be then synthesized properly. And these macrolide and lincolnomide exactly do that. They are like plugs. They put a plug into the tunnel of the protein. Now, what bacteria did is, they don't want this plug. Okay, obviously you don't want an antibiotic to bind. So what they do is, in that tunnel space, they start methylating certain important bases. They can put monomethylation mark like N6 monomethylation or dimethylation. Now, when that happens, then the antibiotic which was binding and acting like a plug can no longer bind, because the methyl group acts like a steric clash. So this enzyme that I'm talking about is the one, once bacteria gets this enzyme, it methylates and does not allow the antibiotic to bind in the same space. And when that happens, then antibiotic can no longer work and bacteria become resistant. So what is our job now? To figure out how to stop this methylation from happening, how to stop the enzyme which methylates. So we looked into this problem, but the problem is, this methylation is very important. There are methyl transferases or enzymes which methylated bases, DNA, RNA, protein. They look very similar to each other. So if I take and try to put an inhibitor for the active site, I will stop my own DNA synthesis from happening. I will stop my own protein methylation from happening. So I have to make sure I make drugs which are only going to inhibit the bacteria and not anything else. For that, we have to find unique places. The way you can find this is, you look at the enzyme. The enzyme does not make a mistake. The bacterial enzyme which methylates and does this reaction only does this reaction. It does not mess up. That means the bacteria knows the information to the specificity to go and do a particular reaction, even if the active site is common, is already there encoded in the enzyme. This small enzyme knows. So we took two such enzymes in the ribosome. One which goes to this position and one which goes to this tunnel position that I'm talking about. ERM is the one which is present in pathogens. This particular one is also a little bit in resistance, but it also is involved in keeping ribosome properly. This enzyme never methylates here. This enzyme never methylates the other place. So they are very, very specific to their particular place. How do these enzymes know they have to go here? And this enzyme knows it has to go here. This is what both the enzymes look like. They look like cousins of each other. If you look, both enzymes look like each other. This enzyme will only methylate at A2058 position. This enzyme will only methylate at KSG. So we wanted to know the targeting determinants. So if we know how they know they have to go there and we stop the targeting, then we will have specific inhibitors. That was our main motivation. So what we did was, we said, let's look at both these enzymes and see if we can convert one enzyme to another enzyme. This enzyme is going at one spot. Now we're going to change the position of this enzyme. Can I convert one to another and figure out what is the targeting determinant? And this is the work we did in this paper, where we show that you can take the protein, change elements of these two proteins. They look very similar, but there are certain loops and small elements which are different. We solved the structure and found out which are the elements which are different. We did established methylation assays so that we can look at inhibition. And then we made many chimeras. What we did was took portions of one protein, pasted in the other, and so on, and made variants of this protein. So what was the job that we wanted to do? The job we wanted to do was we wanted to see if we can take this enzyme, which is methylating ribosome at a different position, and convert it into a pathogenic methylation site. And for that, we made many chimeras. And like I showed you here, we made many chimeras. And in the end, what we figured out was there are two loops, loop 1 here and loop 12 here. If you switch, cut and paste from ERM to KSGA, it starts methylating. That means the targeting lies within these two loops. These two loops decide where the methylation has to happen. It has got nothing to do with catalysis. It has got to do with direction in this case. So here we can show methylation is happening by mass spec, and then we did a lot of catalytic studies. And so we realized where is the actually elements which are happening. So we did this in the test tube, and in the test tube things work much better. So we wanted to test the same thing in the cells. So we took cells where you add erythromycin, and if you do this, then they are sensitive to the drug. Then you add this particular ERM protein, which methylates, and now they become resistant. Then we added a chimera, and if we change the chimera with these two loops and also this head part, we get very nice resistance. So cells, in the context of actually doing it in cells, you need the two loops to be swapped. You also need this head region, which is a regulatory region, so that it can reach the ribosome in the cell. So also, we could also find a site where you can, which is a specific site based on this loop, which now we are using, and we have a grant to work on it to find new inhibitors, which are going to be only for this particular ERM enzyme and not cross-react. Once we have this inhibitor, ERM will stop working, and then we will be able to develop drugs. This all we did from a test tube, and taking a small peptide fragment, DNA fragment, and the enzyme. Now we are working at the level of the ribosome. What we are trying to do is the same enzyme. We are taking the 30S ribosome and trying to understand targeting. So this looks like a complicated picture, but what I want to tell you here is this is the ribosome, and this is where the peptide bond is formed, the active site, and this enzyme goes under this site here and methylates and makes the ribosome whole. Same reaction is happening. And so what we did was to understand the targeting determinants, we solved the cryo-electron microscopy structure of the 30S ribosome with this enzyme. So earlier we were trying to do everything in a small scale with small RNA and the protein. Now we have the whole ribosome, 30S, and the enzyme. And we got many structures from which we can tell a lot about the mechanism of how it is happening. Not only the mechanism, we can tell exactly how the protein is enmeshing inside the ribosome. So you can see this is a cartoon which shows the protein, and the protein is like a ball which is surrounded by ribosome on all sides. And when the protein enters, it rearranges a lot of the ribosome, and so that it can methylate the site of interest where the methylation will happen. So this structure has enabled us to see in a ribosomal context, which is more like how it happens in the cell, how things are happening. Not only that, to facilitate this,
Ruchi, sorry,
almost done. I'm almost done. Just my last slide.
No, I think your slide, we have lost your slide.
Slides are not visible.
You have lost my slides? When did you lose it?
Previous one, I think after that, we lost the slide.
Okay. I will reshare then. I don't know why this has happened. Let me know if the sharing is successful. No?
We can see the slides in the background. Yeah, now we can see.
Yeah, you can see.
Yes. Yes.
Yeah, can you see?
Yeah.
The slides are visible. Yeah.
Okay, so let me. I mean, I was a little worried about the time also. I didn't want to take a lot of time, I did not realize. I'm sorry. So what I was trying to tell you was we were able to solve the cryo-electron microscopy structure of this particular protein, which is a methyl transferase with the 30th ribosome. And what that structure helps you do is it lets you know what are the ribosomal contact points of this particular protein. So this protein, what are the ribosomal contact points and how it methylates, how it rearranges the ribosome so the methylation can happen. So it is shown over here. And in this case, we see that the whole of the ribosomal side, that particular part of the RNA, it flips into the enzyme. And it also tells you how is this protein packaged in the ribosome. To see how important this packaging is, these yellow helix and the blue helix, which is the actual helix, and this orange helix and the green helix, we made variants of the ribosome. So what we did was we made synthetic ribosomes where we can make whatever mutations we want. So in these ribosomes, I can change a base, I can add a segment of my choice, I can remove a segment of my choice, and I can manipulate the ribosome. And after manipulating the ribosomes, I can look at the activity of the ribosome. Is it still methylation is going on or not? So what this tells us is that methylation is very, very, very dependent on the 3D architecture. So the 3D architecture of the site and everything around the protein is very important. Some places like this blue helix is very, very important because that's where the methylation is happening. In other places, you lose partially the activity because the enzyme is no longer tethered properly. So one site can be more important than the other. One can be extremely important. One can be making the enzyme lose. So 50% of the activity will only be there. For example, I talked about the head portion. Head portion, if you remove, then what happens is the activity becomes kind of compromised by 50% because the enzyme is not tethered very, very strongly. So we did many such, this is just shown a few of them, but we did many such mutations to try to understand the biochemical nature of this interaction and how important it is in the ribosomal context, and came up with a similar conclusion that again, for this enzyme, the loops that I showed, unfortunately, the picture I want to go back in 1 minute would be this picture. These loops and the head are very important, even in the ribosomal context, when you are trying to understand what is going on, how methylation is happening, and how it is working in a ribosomal context. With that, I would like to thank the people who contributed. Ju is the person who did the cryo-electron microscopy work, and we did the initial work with Vinod and Rahul, as we are now trying to establish a cryo-electron microscopy center at IIT. But in the meantime, we have been working with NCBS. Ruchika is the former PhD student who worked on our initial templates with the ERM. Unfortunately, this paper was also a part of our work and the efflux pumps. Hussain is the PhD student who had done this, but I could not talk a lot about it in the interest of time. And all the funding agencies have been very generous in helping us through our work. And thank you all for listening, and I'll be happy to take any questions. And I'm sorry if it was a little fast the way I went. Thank you so much.